Hello everyone, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. If you remember when we were discussing about the origin of life, and when we say that okay, these are the potential ways in which the organism could have been originated onto the earth, we talk about the primitive cell, and that primitive cell is being formed within the primordial oceans. So that primitive cell is only the existing species which further got evolved into the first the unicellular organisms and then multicellular organism, and then that got further specialized into the organism with the tissue as well as the organism with the organ systems. So in today's class we are going to start discussing about this particular cell and how the cell is actually functioning, the different types of task, and what are the different organelles are present within the cell and what is the structure of the cell. So when we talk about the cell, as you can see that the higher eukaryotes have multiple organs to perform the specific functions, right, such as liver, kidney and heart, and these organs have the specific tissue and each tissue is composed of the cells, right. So whatever is function, what is happening in a higher organisms like the humans, that it has the different types of organs. Like suppose for example we have the liver, we have the kidney, we have heart, we have the lungs, and all these organs have their specialized functions, but these organs are also made up of tissues, and that tissues are made up of the cell. So function, whatever we say is actually being performed by the cell what is present in that particular organ, and that's why the cell is considered to be the structural as well as the functional unit of the cell. So whatever the function you see from that particular organ could be performed by that particular cell as well, which means a cell is the smallest unit which actually be able to perform all the functions. For example, in a human body we have different types of organs to perform the different types of tasks, like heart is there for circulating the blood, liver is there for detoxifications, kidney is there for the excretion of the byproducts, lung is there for respirations, but the cell, which is actually the structural and functional unit, can be able to perform all these functions on its own because it has all the necessary infrastructure to perform all the functions. Based on the cellular structures, cells are classified either as the prokaryotic cell or the eukaryotic cell. So what you see here is a prokaryotic cell, which is actually a bacterial cell. I have taken the two examples of the eukaryotic cell. I have taken the example of the plant cell, and I have taken an example of the animal cell. So based on the cellular structure, the cells are classified into the prokaryotic and the eukaryotic cell. In most of the cases, prokaryotic cells are the single cell, whereas the eukaryotes are either single or the part of the multicellular tissue system. So before getting into the detail of the structure of the prokaryotic or the eukaryotic cells, let's discuss about the differences between the prokaryotic as well as the eukaryotic cell, so that you will be understand what could be the differences, what is going to happen, and how the eukaryotic cell has evolved from the prokaryotic cell. So what you see here is a table where I have listed the differences. So this is the properties of the prokaryotic cell and this is the property of the eukaryotic cell. Size. Okay, so first criteria is the size, and the size is very small. So prokaryotic cells are mostly in the range of the micrometer range, whereas the eukaryotic cells could be of variable size. They could be up to the 40 micrometer in diameter. So they could be several sizes, RBCs, macrophages, Kupffer cells, and all those kind of things. So they will be very different. As far as the genetic material is concerned, genetic material is circular in the case of the cytosol, and it is present as a free material, which means it is not present in the bound form, whereas the DNA in the form of a linear chromosome present in the well defined double membrane nucleus. So no direct connection with the cytosol. So in the eukaryotes, The DNA is present in the form of a chromosome, and that is present in a well defined structure which is called as the nucleus, and that nucleus is not directly under the contact with the cytosol. Then the replications. As far the replication is concerned, so replication means how you are actually going to make the another copy of your genome, right? So the replication is done by the single origin of replication, what is present in the case of prokaryotes, whereas in the case of eukaryotes it is having the multiple origin of replications. As far as the genes are concerned, so the genes are the functional part of the genome which are actually be responsible for the production of different types of products or different types of proteins. So for the gene there is no intron present, whereas in the case of eukaryotes you have introns are present in the eukaryotes. So don't worry about this particular terminologies, because these terminologies will be clear when we are going to discuss about the replication, transcription and translations in a subsequent modules. Then the organelles, there is no organelle, no membrane bound organelle is present in the prokaryotic system, whereas the membrane bound organelles are with the well defined functions are present. So you have the different types of organelles, you have the nucleus, you have the mitochondria, you have the chloroplast, you have the endoplasmic reticulums and so on. So that all we are going to discuss in this particular module. Then the cell wall, there is a definite very complex cell wall what is present in the prokaryotic system, whereas in the case of eukaryotes, except the fungi and the plant, the eukaryotic cells are devoid of a thick cell wall, which means the animal cells are devoid of the cell wall, whereas the fungi and the plants are going to have the cell wall. Then the ribosomes. So ribosomes are the protein machinery, and they are actually going to be 70S. So this is a kind of a parameter. So that's 70S, whereas in the case of the eukaryote it is the 80S. Then we have the transcription and the translation. So transcription and translations occurs simultaneously in the case of prokaryotic cell, whereas in the case of transcription in the nucleus, so transcription is happening within the nucleus and the translation is happening within the cytosol. As we said already in the beginning that the genome is present in a well defined nucleus, and that is very far away from the cytosol, and the translation is not happening in the same simultaneously. Transcription is happening in the nucleus and the translation is happening inside the cytosol. Let's start about the discussion about the prokaryotic cells. So the simple prokaryotic cell, what is being shown here, right? This is a bacterial cell, right? And the structure of the prokaryotic cell, a simple prokaryotic cell is very simple and smaller than the eukaryotic cell. As we said, prokaryotic cells are in the range of micrometer, whereas the eukaryotic cells are very big compared to the prokaryotic cell. One of the classical difference between a prokaryotic cell and a eukaryotic cell that it has no membrane bound organelle including the nucleus. So what you have is a cell where all the organelles are present within this cytosol, right? And what you have here is the different types of organelle. You have the flagella, you have the genomic DNA or the genome of the bacteria or the prokaryotic cell. Then you have the well defined cell wall, then you have the plasma membrane, and then it has a protective capsule which is actually going to give the strength and protection. And then it has the ribosome, which is called as the protein machinery. Then it also has the food granules and all other kinds of things, and it has the pili, then it has the cytoplasm and the plasmids. So let's discuss about all these the substructures what is present in the prokaryotic cell. So the first substructure is the flagella. So flagella is present in those bacteria which are actually motile. So flagella is present in a bacteria and it is required for the motion within the bacteria. So you can see that if a bacteria is present in a drop, it actually can use this flagella to swim around. So flagella is attached to the bacterial capsule, is a central feature of most of the prokaryotic cell, especially the motile bacteria. It provides the motion or the locomotion to the bacteria, and it is responsible for the chemotaxis of the bacteria. I'm sure you probably are not aware of this terminology which is called as chemotaxis. What is mean by the chemotaxis? Chemotaxis means the attraction of organism, attraction towards chemicals. Okay, for example, if there is a sugar crystal, right, then what the bacteria is going to see, right, it is actually going to move towards this sugar crystal because it is looking for that particular sugar crystal, it wants to eat that, right. So that motion, right, that directed motion of a bacteria towards the particular chemical is known as the chemotaxis. And how it will move? It is actually going to use this flagellum which is attached to the capsule. Movement of a bacteria towards a chemical gradient is known as the chemotaxis. Okay, which means once you have a sugar molecule here, it is actually going to be dissolved into the water, right, and it is actually going to have a gradient. So because this bacteria can be able to have the senses, it can be able to sense this particular gradient, and that's how it will move towards the that particular food source. It could be glucose, it could be any other molecule as well. So flagellum is a part of cell wall and its motion is regulated by the motor protein present inside the cell. So flagellum is attached to the cell wall, and inside it actually has the motor neurons. Like just like in a humans we have the muscles, it has also be attached to the cellular machinery so that it actually can has the motor proteins, and that motor protein actually can change the flipping movement. So the flagellum is actually going to have the flipping movement, right? It's actually going to move like that. And the flagellar motion is an energy consuming process, and it is governed by the ATPase present at the bottom of that particular shaft. It is made up of the protein which is called as flagellin, and the reduction or the suppression of the flagellar protein reduces the bacterial infectivity and ability to grow. So some of the bacteria also uses the flagellum even for accessing the different types of host, and that's how they can be able to use this for reaching to the host, right. So they can also use for reaching to the host, and that's how they can be infectious. So if you actually reduce the production of this flagellin protein, and if somehow you compromise the flagellar movement, you are actually going to make the bacteria non-motile, and that's how the bacteria is going to lose its ability to infect, and that's how they will be not going to cause the disease. Now the second is the bacterial surface layer. So as you can see, the bacteria has a very complex surface layer, because bacteria possesses the three anatomical barrier to protect the cells from the external damages. So since the bacteria does not have the membrane bound organelles, and it is very susceptible for the hypotonic lysis, because bacteria is mostly being present in the water or hypotonic solutions. It has a very well defined anatomical barrier so that it can actually be able to withstand this. So what are the different layers you have? You have a bacterial capsule, which is the outermost layer, and it is made up of high molecular weight polysaccharide. So what you see here is this is the bacterial capsule, right, and this capsule is required because it gives the protections, and this is the outermost layer, and then it is impermeable to the water or other aqueous solvent, and it is responsible for the antigenicity of the bacterial cell. Then you have the cell wall, which is present in the middle layer, right, and the cell wall, and it is giving its response to the Gram staining. And the third is the plasma membrane. So you have the three layers. One is the capsule. So the outermost layer is capsule, then you have the cell wall, and then you have the plasma membrane. And why the bacteria has such a complicated system? Because bacteria is always been present in a harsh environmental condition. It could be present in the water, it could be present in a strong acid solution, it could be present in a alkali solution, it could be present in a solution where a lot of chemical toxicants are present. So because these are things are there, it actually is protecting themselves by using all these layers. So capsule is a very thick layer which actually is not going to allow these chemicals to get inside the cell. Then we have the cell wall. So cell wall composition in gram negative and gram positive bacteria is different. The cell wall has different constituents and be responsible for their reactivity towards the gram stain. So we have the two different types of bacteria. One is called as the gram-positive bacteria and other one is called as the gram-negative bacteria. And both of these bacteria will have the different types of cell wall compositions, and because of that they will be differentially responsible for one of the classical stain, which is called as the gram stain. And because of the gram stain, they are being classified either as the gram positive, which means the gram positive are actually going to give you the staining, whereas they are going to be gram negative if they are not going to be give you the gram staining. Okay, and based on this only the gram staining, the bacteria are classified as a gram positive or gram negative. So let's see what are the different components are present in a cell wall. The outermost layer what you have is a peptidoglycan layer. So this is the peptidoglycan layer what you see here. So the peptidoglycan layer is very thick in the case of the gram positive bacteria. So you see the multiple layers are present in the gram positive bacteria, whereas it is very thin in the case of the gram negative bacteria, and because of this only it is actually having a differential response towards the gram staining. Peptidoglycan is a polymer of the NAG and the NAM. NAG is N-acetyl glucosamine and the NAM is the N-acetyl muramic acid. So NAG and NAM are actually the sugar molecules which are present, and these sugar molecules are connected to each other by a beta 1,4 linkage. And the sugar polymers are attached, because you see this, it has sugar molecules, right? So these are the sugar molecules which are being attached to each other by a beta 1,4 linkage. And alternatively you have the NAM block, then it is connected to a NAG block, and then it has a NAM block, like that. So you see that it has a one layer, then you have second layer, you have third layer, you have fourth layer and fifth layer, and then these layers are being attached by the peptide chains, which are composed of the amino acid L-alanine, D-glutamic acid, L-lysine and the D-alanine, which means it is actually a combination of the L and the D amino acid. These are the two different types of amino acids, and you know that the L amino acids are more abundant in the nature compared to the D amino acids. So the peptide chain present in one layer cross-linked to the next layer to form a meshwork which is responsible for the physical strength of the cell. So what you have is you have the NAM and the NAG blocks, and then the second layer is also having the same way, and then these layers are actually being connected by the peptide chains, and that's how it is actually giving a tensile strength to the cell wall, and that's how they are very robust, or they are very rigid in terms of accepting the outside molecules. And the peptidoglycan synthesis is targeted by the antibiotics such as penicillin, whereas the lysozymes actually degrades the peptidoglycan layer by cleaving the glycosidic bond connecting the NAG and NAM to form the polymer. So you have the two options if you want to destroy the cell wall. What you have is you can actually have the antibiotics. So if you put the antibiotics, what antibiotics is going to do is it is actually going to target the peptidoglycan synthesis. One of the classical example is the penicillin. The other option is that you can use an enzyme which is called as the lysozyme, and that lysozyme is actually going to degrade the linkage between the NAM and the NAG. Lysozyme is a very important enzyme what is present in our tears and as well as the saliva, and that's how the tear and the saliva are actually protecting the humans from the bacterial infection, because as soon as the bacteria goes, irrespective of whether it is a gram positive bacteria or the gram negative bacteria, a lysozyme is actually cleaving the bond between the NAG and NAG, and that's how they are actually destroying the cell wall. And once they destroy the cell wall, these bacteria are very susceptible for the osmotic damages. So they will be very susceptible for the water, or they will be very susceptible for the tear like conditions, and that's how they are actually going to get lysed, and that's how they will die. So this is one of the strategies, and that's how people are trying to develop many antibiotics which are actually going to work on the peptidoglycan synthesis. Apart from that, the cell wall is also having the lipoteichoic acid. So apart from the peptidoglycan layer, you also have the lipoteichoic acid which is present in the cell wall. So lipoteichoic acid are only present in the gram-positive bacterial cell wall, and it is an important antigenic determinants. Okay. So that's why this for lipoteichoic acid, our immune system is actually going to work, and that's why it is actually going to produce the response. Then we have the lipopolysaccharides or the LPS. The lipopolysaccharides are only be present in the gram-negative cell wall, and it is important antigenic determinants. So compared to the lipoteichoic acid, which is only present in the gram-positive bacteria, in the gram-negative bacteria you have the lipopolysaccharide, and that lipopolysaccharide is a very important antigenic determinant, because that is actually going to induce the immune response in the humans. This is, I have just given you a write up so that if you are interested, you can actually be able to read about the gram staining. So gram staining is a staining which is been developed by a Danish scientist, which is called as the Hans Christian Gram. And as I said, gram staining is gram-positive bacteria is taking up the gram stain, whereas the gram-negative bacteria are not taking up the stain. Be more interested about reading the gram staining, you can be able to go through with this publication, and as well as I have given you a small write-up so that you can also go through this particular write-up as well. So now let's move on to the beyond the cell wall. So apart from the cell wall, they have the cytosol and the other organelles. So prokaryotic cells do not contain any membrane bound organelle. The organelles are present in the cytosol, such as the ribosome, which is the 70S ribosomes, and the genetic material, whereas the electron transport chain and complexes are embedded within the plasma membrane. So within the plasma membrane you have the electron transport chain. You will see the description about the electron transport chain when we are going to talk about the mitochondria. Apart from that, the genomic material is present in the chromosome and as well as the extra chromosomal DNA. So prokaryotic cells contain the genetic material in the form of a circular DNA known as the bacterial chromosome. So bacterial chromosome is different from the eukaryotic chromosome what is present in the eukaryotic cells. It contains the genetic elements for the replications, transcription and the translations. Bacterial chromosomes follow a rolling circle model of the DNA replications. The genes present on the chromosome does not contain the non-coding region, which is called as the introns, and it is coarse translated to the protein. Besides main circular DNA, bacteria also contain the extra chromosomal or extra circular DNA known as the plasmids. So what you see here is actually a plasmid. These plasmids are called as the extra chromosomal DNA, which means they are actually important for the bacteria, but they are being present as a extra chromosomal DNA. Presence of plasmid containing resistant genes confer the resistance towards the known antibiotic. Exchange of extra chromosomal DNA between the different bacterial strain is one of the mechanisms responsible for the spread of antibiotic resistance across the bacterial populations. So the plasmid is very important because the plasmid is the only genetic material which actually been exchanged between the different bacterial species, and that's how they can be able to exchange their properties with the neighboring residues, right? For example, if a bacteria has suppose 200 copies of a plasmid which is actually giving the resistance against the antibiotic, for example antibiotic penicillin, right? Then what it will do is it will actually going to give some of these plasmids to another bacteria which is actually sensitive bacteria. So once these sensitive bacteria are actually going to receive these plasmids, they will also going to be resistant for the antibiotics. That's why it is important that when people are working in the laboratories or when people are working in biopharma industries or something, these plasmids has to be the plasmid bearing bacteria which people are generating while they are doing the recombinant DNA technology has to be destroyed very nicely, so that the genetic pool of this plasmid should not go into the environment, and that's how there will be an exchange of genetic material or exchange of the plasmid between the two bacteria. It is actually going to spread the antibiotic resistance even in the natural bacteria, and that is one of the mechanism through which the bacteria are acquiring resistance, and they acquire the resistance very fast because the exchange of the plasmid material, and that's why it is important to study about the plasmids. So plasmid is a circular DNA, and there are different forms of plasmids what is present when you are going to do the plasmids for the different types of treatment. For example, so you have the bacterial plasmid is a double circular DNA molecule, and it exists in the 3 different forms. If the both strands of the circular DNA are intact, it is called as the covalently circular DNA. So this is what you see here is a covalently circular DNA. Whereas if one of the strand has nick, then it is acquired the conformation of the open circular DNA. So if you are actually going to put the nick in one of the strands, like for example here, then it is actually going to acquire another conformation which is called as the open circular DNA or the OC form. This is called as the triple C form, this is called the OC form, and the third is called as the supercoiled form. During the isolation of the plasmid DNA from the bacteria, covalently circular DNA loses few numbers of turn, and as a result it acquires the supercoiled conformations. Interchange between these forms are possible under the in vitro or the in vivo conditions, such as the DNA guidase produces the additional terms into the circular DNA to adopt the circular conformations. So bacteria plasmid is actually acquiring all these 3 conformations under the in vitro or the in vivo conditions, and that's why they can be, the different enzymes are working. For example, if you take the circular DNA and if you put the DNA guidase, it is actually going to create the turns into this, and that's how it will going to generate the supercoiled DNA. But if you take the supercoiled DNA and treat it with the topoisomerase, it is actually going to reduce the turns, and that's how it is going to be turned into the closed covalently closed circular DNA. Let's see one of the plasmids. These are the bacterial plasmids which are very commercially being available or very being used in the recombinant DNA technologies. So plasmids are widely being used for the cloning of foreign DNA into the bacterial system as a host strain. And this is the plasmid which has the different types of components. One of the thing what you have here is the origin of replication. So origin of replication is a place from where the bacteria is actually going to start its replications. Then it also has the antibiotic resistance genes. For example, here you see it has the antibiotic resistance genes which is for the ampicillin. So if this plasmid will go to any bacteria, it is actually going to give the resistance against the ampicillin, the antibiotics. So the antibiotic resistance genes or the enzymatic gene is responsible for giving the phenotypic changes in the host after the entry of the plasmid. Apart from that, what you see here is an enzyme which is also being present within the plasmids, and because the plasmid has the origin of replication, it has the antibiotic resistance, and it has all these components, they are independent, and that's how they are very dangerous, because they can be independent, they don't be dependent on the nucleus for its replications or early activities, and that's why they can independently go to the new bacteria, and the new bacteria is also going to have the additional features whatever is this plasmid is actually acquiring. Let's see how we can be able to isolate the plasmid from the prokaryotic cell. The plasmid isolation is a multiple step process. It is having many steps. So in the step 1, what you have to do is you have to collect the bacterial. So first you have to do is you have to take the bacteria, you have to transform that bacteria with the plasmid, or suppose the bacterial plasmid is present in the bacterial cell. First thing is what you have to do is you have to grow these cells so that you have a sufficient number of bacteria. Then you have to do, in the step 1, you have to do a centrifugation, and then you have to resuspend these bacterial cell in a solution 1. The solution 1 is actually containing the 50 millimolar glucose, 25 millimolar tris HCl, and 10 millimolar EDTA. So the method what we are discussing is called as the alkaline lysis method. In the step 2, you are going to do the alkaline lysis. So in the step 2 you are going to do the alkaline lysis. That alkaline lysis you are going to do with the help of the 0.2 normal NaOH and 1% SDS, and that is actually going to lyse the cells and it's going to denature the DNA. Ultimately you are going to do the third step, which is called as the renaturation. So in the renaturation is going to be performed by the potassium acetate solutions and the glacial acetic acid solution. And what will happen is that in this step there will be the renaturation. So that renaturation is actually going to renature the plasmid DNA, but it will not renature the chromosomal DNA, and because of that the chromosomal DNA is actually going to be discarded in the next step. So when you are going to do the centrifugation, the chromosomal DNA, since it is not been renatured, it is going to be precipitate and it will going to be present in the form of pellet, whereas the supernatant is going to contain the plasmid DNA. Then in the step 4 you are going to just do the purification of this plasmid. So you are going to do the deprotonations, and that will result into the isolation of the plasmids. That deprotonation you are going to do with the help of the chemical which is called as the phenyl chloroform isoamyl solutions, and that is actually going to remove the protein so that you can be able to make the very high purity plasmid DNA. And in the step 5 you are going to resuspend that plasmid into a alcohol from the solutions. So that's what you are going to get. In the step 4 you are actually going to have the plasmid as well as the protein, and then what you're going to do is protein will be present in the precipitate. That supernatant you are going to collect, and then that supernatant is going to add the alcohol, and once you add the alcohol the plasmid is going to be precipitate, and that's how you are going to isolate the pure plasmid. And that pure plasmid can be used for the different types of applications, like different types of applications which we are not going to discuss. So let me give you a very real experience how you can be able to isolate the plasmids from the bacterial cell. So I'll take you to my lab where my student is actually going to show you a very small demo and how you can be able to isolate the plasmids from the bacterial cell.
Hello everyone, in this video we will show you how to isolate plasmid DNA using alkaline lysis method. For preparation of plasmid DNA, we need resuspension buffer, lysis buffer and neutralization buffer. In addition to that, we need isopropanol, RNase A and ethanol. Resuspension buffer contains 25 millimolar tris and 10 millimolar EDTA, and we have to add RNase A at a final concentration of 100 microgram per ml. Lysis buffer contains 0.2 normal sodium hydroxide and 1% SDS. Neutralization buffer contains 3 molar potassium acetate pH 6.0. For isolation of plasmid DNA, we need at least overnight grown culture with OD of 3.0. So this is already a cultured one. We have to harvest the cells by centrifugation. These files we have to centrifuge 11,000 rpm for at least 1 minute to get the cells precipitated. Now we got the cell pellet. We can proceed for alkaline lysis method to isolate plasmid DNA. In first step, we are going to add resuspension buffer which contains RNase. Mix thoroughly until all the cells suspended in resuspension solution. After the cells got suspended completely, now we have to lyse the cells using strong alkaline condition, that is 0.2 normal sodium hydroxide and also 1% sodium dodecyl sulfate. Now we have to gently flip the tube in order to lyse the cells completely. We can keep in this condition for up to 5 minutes but not more than 5 minutes, which will degrade the plasmid DNA and also genomic DNA will come out and it will interfere with the mini-print. In next step, we have to neutralize the sodium hydroxide using neutralization buffer to prevent any further degradation. After adding neutralization buffer, you can see there is a white precipitate. That means all the proteins precipitated by neutralization buffer. You can flip the tube 2-3 times, completely precipitate all the remaining proteins. Now the solution part contains our plasmid DNA and all the precipitated one contains genomic DNA and also the proteins from bacteria. Now we have to centrifuge this lysate for 10 minutes at 11,000 G. The precipitate got settled. Now we have to transfer the white clear supernatant to another tube. This clear supernatant contains plasmid DNA. Now we have to precipitate this plasmid DNA with the isopropanol, followed by washing with the 70% ethanol. We can see white precipitate in the solution. Now we have to centrifuge it, collect the white precipitate and wash with the 70%. After precipitating plasmid DNA with the isopropanol, we will get a pellet of plasmid DNA. Now we have to wash that pellet. We wash this pellet with the 70% ethanol. Again centrifuge the pellet. Now we got the pellet. We have to air dry the pellet and dissolve it in deionized water or tea buffer. We'll leave at room temperature till the ethanol got evaporated. Next we will add tea. To ease the process of manual alkaline lysis method, there are several kits available from commercial vendors. The basic difference between alkaline lysis method and the kit based method is, kit based method contains silica based columns where lysate which containing plasmid DNA binds to these beads, and after washing whatever the unwanted components are, they will elute out and we will elute the plasmid DNA in TE buffer or water. The composition of the lysis buffer is same as previous method, and also neutralization buffer, resuscitation buffer, every buffer contains same composition. But in commercial kits we have one extra wash buffer which will remove any unwanted contamination and give pure DNA.
So when you see this demo, what you could see is that the students was discussing about all the 4 or 5 steps what we have just discussed, and after these steps what you are going to get, you are actually going to get the plasmid like this. So what you see here is the 3 forms of the plasmid. You have the covalently closed circular DNA, which means the CCC forms. You are going to have the OC forms, and then you also going to have the supercoiled form. So what you see here is, this is actually the covalently closed circular DNA, this is the open circular DNA, and this is the supercoiled DNA. And whereas since we have not used the RNAs, you are also going to see the some amount of RNA, because RNA is also present in the bacterial cell. So what we have discussed so far, we have discussed about the bacterial prokaryotic cells. We have discussed about the structure of the prokaryotic cell. We have discussed about how the cell wall is. Cell wall is thick in the case of the gram-positive bacteria, whereas it is thin in the case of the gram-negative bacteria. Apart from the cell wall, you also have the capsule and as well as the plasma membrane, and all these barriers, anatomical barriers, are making the bacterial cell very resistant for the environmental changes or the chemical what are present in the environments. So with this, I would like to conclude my lecture here. In our subsequent lecture, we are going to discuss about the eukaryotic cells. Thank you.